In the last 3 lectures, I've covered the structure of folded proteins, folded globular proteins. And today I'm going to be talking about the structure or the lack of structure of disordered proteins. My background in this work is related to my work with Liz Rhodes, who was a professor in MB&B at Yale, and now she's in chemistry at University of Pennsylvania. And we wrote a series of papers in the mid 2010s on structure of intrinsically disordered proteins. And Wendell Smith, a physics graduate student, worked on these projects. So some initial thoughts on protein aggregation. So the cartoon in the top of this slide suggests that when proteins are folded into their native structure, they don't aggregate and stick together as much as if, for whatever reason, a protein doesn't fold, and then hydrophobic residues are not buried into the cores of proteins, and then those unburied hydrophobic residues that are on different proteins can bind together effectively and form a nucleus of an aggregate, which can then continue aggregating to form a protostructure. In this case, the intrinsically disordered proteins are going to aggregate into fiber-like structures, and so then form amyloid, which in this case is a twisted fiber. So the protein aggregation in the context today we're going to talk about related to various diseases, for example, neurodegenerative diseases. And the basic idea I've already suggested is that for different reasons, the hydrophobic residues are not buried in the core of a protein. They're exposed to solvent or exposed to other proteins, and then those structures can aggregate and then form larger and larger aggregates. So some of the reasons why proteins would be transiently unfolded could be that particular amino acid sequences don't lead to very stable folds. But you can also get unfolded structures by varying the pH, the temperature, the solvent conditions, and other aspects. So some terminology, so amyloid fibrils can occur in vivo, they can occur in vitro. For example, many of the in vitro assays involve polypeptides, and those polypeptides, essentially, most polypeptides can form aggregates with sufficient chemical denaturants. But amyloid fibrils are typically rich in beta sheet structure, and they're ordered, as opposed to various inclusion bodies or disordered aggregates where they're not forming ordered substructure. So, for example, some pictures of amyloid fibrils on the left is a microscopy image The scale bar there is 100 nanometers. And so this is an electron micrograph of fibrils that are formed from a particular domain of a protein called the SH3 domain. And you put it in solvent conditions at low pH, and they will slowly form these fibrils that form a gel. And it's a little more difficult to determine the substructure within the fibrils. So on the right is a model of what they could be. So for example, each SH3 domain forms a beta strand or beta sheet structures in each of four, what do they call them, protofilaments that then wrap around each other to form a full fibril. And so the basic idea here is that these fibrils can form, for example, at low pH with a whole host of different types of proteins and protein subdomains, as long as the conditions are favorable for that. So sort of non-physiological conditions. And then the idea is many of the structures form interesting microscopic structure. For example, amyloid fibrils are rich in beta sheet content. And so that was in vitro, and here are some in vivo images from, I think most of them are diseased brains. And so protein aggregation is responsible or is connected to many neurodegenerative diseases, Huntington's disease, Alzheimer's disease. And the aggregation occurs in different proteins, as you'll see in these slides. So in panel, so for example, in panel B, there are these Lewy body inclusions, which mean sort of disordered aggregates. And they contain alpha-synuclein, which is a type of protein that we're going to talk about that has a tendency to form intrinsically disordered structures. Well, that it's in an individual alpha-synuclein protein is often disordered and then can form these aggregates. And there are related structures in the synuclein family, beta and gamma synuclein, that also form aggregates. And then in panel E, there's an Alzheimer's disease sample taken from a brain with Alzheimer's disease, and that has another protein, which is a protein called tau. And then in the final panel F, there's another Alzheimer's disease sample from an Alzheimer's disease cortex with another protein called amyloid beta that tends to form amyloid structures. So in each case, the particular protein that's targeted, they'll set up a stain for it, and then the aggregates are these dark images from these samples from various neurodegenerative diseased brains. So again, the basic idea for intrinsically disordered proteins is for reasons that we're still looking into, the proteins don't have a folded state in these particular environments. It's not completely clear that whether these microenvironments are really sort of non-physiological, that there's local deviations in pH or sort of cellular crowding, but these proteins are somehow not very stable. And so they exist in a wide variety of states, not, for example, in the cartoon of globular proteins, the cartoon was that the protein was always folded in physiological conditions, had a well-defined structure, and that structure was connected to its function. In the proteins we're talking about now, they can fluctuate. Sometimes they can be fairly compact and collapsed, and other times they can be very extended. So we're interested in studying that. One important issue on the computational side is what level of modeling do we need and will we actually use? And so I remind you of the different levels of faithfulness in the modeling. So on the left is an all-atom model of an intrinsically disordered protein. So this is alpha-synuclein, which I'll tell you in more detail, exactly how many residues. It's roughly in the order of 100. I'll tell you its exact amino acid content, but you can model it on the left in all atom detail, including the hydrogens. In the middle is a extended atom model where you get rid of the hydrogens and you subsume them into the heavy atoms that they're associated with, so like a nitrogen or a carbon. And then at the coarsest level, you have what's called a C-alpha model, where each amino acid is represented as a single spherical monomer. And there are also hybrids between the most coarse that I represent on the right and then an extended atom. You could also do a C-alpha model where you have a single C-alpha atom for each amino acid, but then you have one or two atoms representing the C-beta or additional side chain atoms. So in general, what we're going to be talking about are simulations of these proteins that are modeled like polymers, either at a very coarse level where you have one sphere per amino acid, or whether you have all atoms included in the amino acid. And so here's an example of the coarsest representation. And so here, this particular simulation is was a cooling. So it was taking a protein at the highest temperature and then running a cooling protocol down to very low temperature. And so here you see that what happens during cooling is that in general, polymers will collapse and form these very compact structures. And so one issue with intrinsically disordered proteins is to try to understand what temperature does that collapse occur at. If it's a very low temperature, then the protein is going to be intrinsically disordered. It's not going to be collapsed at physiological temperatures. And if that collapse temperature is very high, then at physiological temperatures, it's going to be in a collapsed folded state. So a common technique experimentally for understanding structure is what's called SM-FRET, or single molecule Forster resonance energy transfer. And this technique allows you to determine the distance between different residues by using a pair of fluorophores. So what you can do is shine a laser on the system, and that will cause one of the fluorophores. So the fluorophore will be a pair, And so one of the fluorophores will be on one of the amino acids, and then another fluorophore will be on another amino acid, and between which you're trying to measure the separation between them. So you imagine a pair of fluorophores, one on one of the amino acids, one on the other. And one is a donor, which will be excited to emit a photon. So for example, you can excite it to emit a photon in the green part of the spectrum. And then if the other fluorophore is close to that residue, it will accept that. It will receive that photon and then be excited and emit a separate photon in the red part of the spectrum. And then what you can do is that the efficiency or the degree to which the second fluorophore accepts the green photon is determined by this fret relationship. So E fret is equal to 1 over 1 plus r to the 6 over r naught to the 6th. And so if they're very close together so that the separation between the fluorophores is 0, so capital R goes to 0, then the fret efficiency is very high. So this number, E fret, goes between 0 and 1. So if they're very close, the second fluorophore is going to feel the excitation from the green photon and then emit a red, and you can measure the amount of red photons that that are occurring in the system. So that's why when the two fluorophores are very close together, you get a high fret efficiency, and if the fluorophores that are on the target residues are very far away, you're mainly measuring green, there's not a lot of red being emitted, and you get a low fret efficiency. The R0 is a characteristic distance which is particular to each fret pair, and that just is a parameter in the fret efficiency. So at the time we were doing this work, Liz Rhodes had done most measurements. So the idea is that you have to make these fret pairs separately and put them on individual instances of the protein, sort of one by one. So you can have a pair of fluorophores that's at, residue 54 and residue 72, residue 72 and residue 92 on alpha-synuclein. And so she had engineered this system where she had done 12, it's 4 times 3, fret pairs on different residue pairs. And so you do want the most pairs. So the number of pairs you can have is obviously the number of residues times the number of residues minus 1 divided by 2. So roughly sort of n residues squared pairs. And the more pairs that you have that you can measure, the more structural information you'll have about this fluctuating protein. And so 12 was the state of the art about 5 or 8 years ago. And so in prior work, there were sort of rules of thumb about determining whether a protein was intrinsically disordered just based on its sequence. And so some of the parameters that people looked at were the total charge and the hydrophobicity. So Q is the total charge. Each residue will have a charge that I called Q sub I. And so you sum that up over the number of residues. In this case, I take the magnitude and then I divide by the number of residues. And so that's the average charge of a protein. In this case, the charges can cancel. So you could have one residue that had a positive charge, another residue that had a negative charge, and that could cancel. So another thing to look at would be to square the charge, because even though charges cancel, they can have longer range effects, for example, dipoles. And then hydrophobicity, we're going to talk about it more in a couple of slides, but the hydrophobicity, at least the way I'm thinking about it, is a number between 0 and 1, where 0 is very hydrophilic, so non-hydrophobic, and 1 is very hydrophobic. And you can think of hydrophobicity as the likelihood that a particular residue is going to be in the core of a protein, of a folded protein. So for example, an alanine or a leucine has a very high hydrophobicity. They're very frequently found in the core, whereas the charged residues like lysine and arginine have low hydrophobicities. The way that I get those numbers will be discussed more in a few slides, but one way to do it, at least in a sort of data-centric way, would just be to do what I just mentioned, would be to find the probability that a particular residue occurs in a protein core or folded proteins. So in prior papers, so what I show in this table are data from proteins, from amino acid sequences. So in the blue are proteins from the protein data bank that have well-folded structures. So I just give the data point an HQ value from its sequence. And the green circles are data points from known intrinsically disordered proteins. And then I have some other data points in like the big symbols. There are triangles that are colored, and then there's circles that are colored, and then there's a big black X. And so those are proteins that I'm going to refer to later on in the slides. And so I just wanted to highlight those. So the first one is lysozyme. So that's a known folded protein, and that occurs to the right, right lower side of the diagram, which I want to highlight. That would be the high hydrophobicity side and the low total charge side. So that makes sense for folded proteins. They're not typically overcharged, and they're typically hydrophobic. And that causes them to be very stable in their folded structure. Whereas on the other side, say upper left, you have low hydrophobicity and high charge. And since it's high charge, the charges want to separate from each other. And so that in general leads to extended structures. And so the upper left moves more towards the intrinsically disordered side. And so I don't know if it's fully borne out in the data that I show, but there are definitely other papers that have shown this line that goes to the right with a particular slope, and that separates intrinsically disordered proteins from folded proteins. So don't take that as a theoretical rule. Just take that as sort of a rule of thumb that folded proteins are on the lower right and intrinsically disordered proteins are more probable as you move to the left and up. And some of the ones that we're going to talk about more in later slides are sort of on the edge between intrinsically disordered proteins and folded. That's like the green triangle, which is gamma-synuclein and alpha-synuclein and beta-synuclein. But some of the others like pro-T alpha or well, that's the main one that I'm looking at right now, is definitely within the, towards the high charge, low hydrophobicity side. Okay, so So we just talked about some of these proteins in the previous slide. I just wanted to formally give you their amino acid compositions and give you their sizes. So yeah, so MAP. Okay. So MAPT, I'm going to sometimes call tau. So the capital T at the end stands for tau. So I call that tau. It's the largest protein that we considered. It has 440 residues. The synucleins have around the same number, between 127 and 140 residues. And Pro-T alpha is a little bit shorter. And so these all are intrinsically disordered proteins under various conditions. So you can coax them into being intrinsically disordered, and then they can form aggregates. And we'll see in a moment. So here you see their amino acid content. Yeah, so I'll highlight that content basically now. So what I'll do is, to talk about their amino acid content, I'll talk about their charge and their hydrophobicity. So the charge is Q sub I and the hydrophobicity is epsilon sub I. And I'll do that by contrasting it with a folded protein. So the dashed black dots are for lysozyme. And so in terms of hydrophobicity, the hydrophobicity for that folded protein is 0.5 or above for every residue. So it's a very hydrophobic protein. In particular, it can have residues, so it doesn't really go below 0.5, and then it can have residues that go up to 0.8. And then if you look at the charge, it's just not very charged at all, so most of it's at 0, and then there's a little bit at the end that has a slight bit of charge. Whereas if you compare that to the others, the synucleins, the tau protein, and Pro-T alpha, they're mainly charged and mostly negatively charged. So you see the purplish blue, the dashed orange, yellow are very highly charged. And then the tau protein, the one that's very long, it's not very highly charged, but since it's so long, it has these deviations from 0 that are around 0.1. And so if you sum that up over, a wide range, you get a significant amount of charge. So it's mostly positively charged on the large residue side, and then it has some negative charge at the low residue side. The other thing for the hydrophobicity is that they're mostly 0.5 or below. So for example, the tau protein doesn't really have many residues above 0.5. They're mostly around 0.4. And then the orange dashed line has even fewer hydrophobic residues. So in general, the rule of thumb, as I said before, is that folded proteins have low net charge, high hydrophobicity. Intrinsically disordered proteins have the opposite, tend to have significant charge and low hydrophobicity. So what we're interested in doing is that I mentioned that Liz had developed all this fluorophore chemistry where she can put lots of pairs of fluorophores sort of all over the protein. And she's going to be measuring separations to try to figure out what the internal structure of these fluctuating proteins are. And I need to develop a model that can basically recapitulate those inter-residue separations. And so in this lecture, I'm mainly going to talk about the coarse-grained model, where I'm modeling every amino acid as a single spherical monomer. So each monomer will be a sphere. Each amino acid will be a sphere. It'll have a mass. It'll have a diameter. It will have a hydrophobicity. It will have a charge. The separation between these monomers is just based on stereochemistry by adding up basically the peptide bonds. So it'll be at 3.9 angstroms, and there's a typical bond angle between those C alpha atoms. And so in this coarse-grained model, I'm going to have bond lengths which link together the C alpha atoms. There's going to be a bond angle between successive triples of amino acids. And so that's the stereochemistry of this model. When we were talking before about folded proteins, I didn't do any dihedral angles because I talked to you many, many times about that the dihedral angles can be obtained, just by implementing the stereochemistry. But since I'm using a coarse-grained model, the monomers don't have realistic shapes. They're just spheres. I can't get the dihedral angles just from the geometry of the amino acids. So I have to put in a dihedral angle potential that will mimic what's seen in proteins. So I add in this dihedral angle potential, and I'm going to try to fit the coefficients, the A's and B's, to those that are ideally found in experiments. Then I'm going to have a repulsive part of the potential between non-bonded monomers so that the monomers don't overlap each other. So that's just based on their sizes. So like monomer 4 can't overlap monomer number 1. And the overlap is based on the relative sizes of monomer 4 and monomer 1. And then I have an attractive Leonard-Jones potential, and that's supposed to mimic the hydrophobicity of the amino acids. So that's going to be related to the relative hydrophobicities of the monomers. And then the next thing is an electrostatic interaction, which is screened, and it's using a screening length that's typical for physiological conditions, 9 angstroms. And so I used a screened Coulomb potential. That's based on the charges of the monomers, the screening length, and the typical size of each monomer. Now, how many parameters do I have in this model? So the KL for the bond length can just be determined by protein structures, so it can just be determined by the fluctuations that I see in the bond lengths. Same thing for the bond angles. The K theta is just determined by what's found in experimentally determined protein structures. I already mentioned the dihedral angle potential. Those are experimentally determined. There's not much wiggle room in the repulsive part of the potential because I don't want overlaps between atoms. So the only parameters really are the overall coefficient of the attractive interaction and the coefficient of the electrostatics. And so that's going to be the ratio that I'm going to use to try to compare to experiments, the ratio of attraction to the electrostatics. I'm going to call that parameter alpha. So I mentioned before that I don't put in dihedral angle interactions in the folded proteins because those are determined by geometry and because the model was an atomic model. But I need a dihedral angle potential in the coarse grain model so that I can, specify the structure of the protein. And so what we did is we carried out a sort of a first simulation with no electrostatics and no attractions. So just like a random walk simulation of the proteins, just to measure the dihedral angle potential that the dihedral angle distribution that you would see in a randomly fluctuating protein of each type. And what that is the red and green distributions on this plot. So it tells you basically how likely are you to see a particular dihedral angle among the, each quadruple of C alpha atoms. And so there's a lot of content sort of at negative 180 and not much at 60. So in the background, I also show these dihedral angles that are obtained just from folded proteins for these sort of coarse grained dihedral angles. And you see that the distribution from the intrinsically disordered proteins that are just fluctuating come mainly from beta sheet content, which is the purple-blue part of the distribution, and the alpha helical content is at the orange. And so there's a little bit of a plateau, but not too much structure. So that it says that the dihedral angle distribution of a randomly fluctuating protein mainly comes from the beta sheet content. And so what we can do is we can enforce that dihedral angle potential so that it matches this distribution in our subsequent simulations. So basically, we have a dihedral angle potential that looks like the red curve. So here's a plot of the interactions that we're using, the repulsive, the attractive, and the charged interaction. So on the left panel, panel A, is the repulsive potential. And so what this does is it's flat if non-bonded atoms are not overlapping. And then if they overlap just a little bit, there's a huge repulsive force that kicks in when they overlap. Then there's an attractive interaction. That's the red line in the middle. And so there's no forces at small separations. And then as you go a little higher, you see that curve. And the way you find out what the forces are is you take the derivative of the red curve. So on the panel A, all these derivatives were negative. And so that indicates, they're pointing from upper left to lower right, all the slopes. And so that means a repulsive force. Whereas in panel B, if you look at the slope of the red curve, the slopes are positive. And so that means an attractive force. And so in panel B, I've added a dashed line for the repulsive force, dashed blue line, and then the red part represents the attractive forces. And so you see that it forms a well. The repulsive forces on the left and then the attractive forces on the right form a nice well where the system likes to sit at the energy minimum. Then you can add to that in panel C the charged interactions. And you see that for charges with different signs will have an attractive interaction, and that's why you see that the slope for this interaction are positive. And if you had same charges, the sign would flip and it would be on the upper side and it would have negative slopes. So this charge interaction could be positive or negative depending on whether your charges are the same sign or different signs. With that said, it is hard to distinguish the repulsive interaction from the charged interaction because they're both repulsive, and on the same token, for the attractive interaction with it's hard to distinguish the attractive interaction with the charged interaction with opposite signs because they're both attractive. And so that discussion that I just said will come up in a few slides as well. So this slide indicates the hydrophobicity index. So the hydrophobicity index goes between 0 and 1 to a large degree, or you can come up with a hydrophobicity index that just talks about the relative probability for a residue to be within a protein core. But other researchers have made other definitions. So here I list 7 examples. So Kite Doolittle, Monera, Wimley White, Eisenberg, and so on. Those are all the different symbols. And they've all been normalized so that they go between 0 and 1. And so since I normalize them all between 0 and 1, I can find an average over those 7 hydrophobicity scales that goes between 0 and 1. And I've ordered the amino acids on the horizontal axis according to that average so that it's always monotonically increasing. So you see the very the non-hydrophobic residues are arginine and asparagine, so on and so forth. And then the very hydrophobic residues are isoleucine, leucine, phenylalanine, and so on and so forth. And so this is a way where I can apply specific hydrophobicity scales to specific residues. So then the goal is to, I then basically defined my model. It has bond lengths, it has bond lengths in the sense that you see that this simulation, all the bonds are maintained. It's stays connected. I've included bond angles so that you see that there's some bend in the chain, which corresponds to proteins of known structure. I have the dihedral angle potentials in there so that every 4 residues matches proteins of known structure. And then I have the repulsive interactions in there. So if I slow it down, you can see that the monomers are not are not passing through each other, that it's a chain. And then there are the various attractive interactions and repulsive interactions caused by charge and by hydrophobicity. And so this is alpha-synuclein, and so it's definitely not collapsing and then staying collapsed. And it's definitely, there are periods, we just went through one where it's very extended. And so in general, it's complicated that this protein can take on very collapsed states, but then it can also take on very extended states. And so what we're going to do is measure the separation as the system is fluctuating. We're going to be measuring the separation between all different pairs of residues, and in particular those residues that have been the separations between pairs of residues that have been characterized in experiments. And so here's some of the data, both from the simulations and the experiments. Here I'm showing 4 intrinsically disordered proteins, alpha-synuclein in the upper left, beta-synuclein in the lower left, gamma-synuclein in the lower right, and then pro-T-alpha. Pro-T-alpha, we only have 2 residue pairs, but you see for alpha-synuclein, we have those 12. And so what I'm doing is showing you the FRET efficiency. You saw that there was a direct mapping between the FRET efficiency and the separation between monomers. So I can calculate a FRET efficiency, for the simulations, and obviously for the experiments, they just measure directly a FRET efficiency. The experimental data will appear in the black symbols for each protein, and then the models occur in the color, so the red and the purple, blue with different symbol types. There are 3 types of models, but it's really only think of 2. One, which is the lower, I think they're diamonds. So just consider the lower diamonds and the the red circles, or they're squares, just the red and the lower purplish blue. And so first, with the experimental data, the way I organized it from left to right on the horizontal axis is from low separation on the left to high separation on the right. So like for alpha-synuclein, we have the numbers 54 to 72. That means those are the residue ID numbers. So they have a separation of 18 residues. So that's a relatively small separation. Whereas on the right, I have 9 to 130. So that's a separation of 121. To convert residue separation to distance is one of the main goals of this study and in general protein structure, but a rule of thumb is that the distance scales as the separation to the 1/2. So 18 to the 1/2 versus 121 to the 1/2. So the square root of 18 or the square root of 121 is how you map it to a true separation between residues. But in general, since I've organized it from small separations to large separations, moving from left to right, you agree that the FRET efficiency should go from 1 to 0, because at small separations, the FRET efficiency is very good, very high, 1. But then as the residues move away from each other, the FRET efficiency gets smaller and smaller. So there are some fluctuations on the way, but in general, that trend goes from 1 to around 0.4 for the case of alpha-synuclein. And so then the 2 models I want you to consider are the one model, which I want to highlight is not a very good model. It's one that doesn't have any attraction. So the purplish data is just an electrostatic model. So since we know that intrinsically disordered proteins are fairly charged, and of the same charge, either exclusively positively charged or exclusively negatively charged, you see that would tend to extend the protein, which would lead to smaller values of the FRET efficiency. And so that's why the charged model doesn't fit. But then when we have the red model, where we're able to sort of tune the ratio of the attractions to the repulsions, we can get basically quantitative agreement with the experiments. And that's definitely true for alpha-synuclein. It's true for beta-synuclein. It's true for gamma-synuclein. Well, it's true for all 4 of them. And again, for all 4, the electrostatics model occurs below the rest, because it's basically showing that there is some attraction, there is some hydrophobicity in these intrinsically disordered proteins that make them different from just a charged polymer. Just want to show you the other protein tau, which is a longer protein. Basically the same conclusions there. The experimental data is in black. The red is sort of the best match between, the best balancing between hydrophobicity and charge. The charged model which is the diamonds, is well below the experimental data. So again, intrinsically disordered proteins need a balance of hydrophobicity and charge. Just to show you that sort of how we figured out the right balance between hydrophobicity and charge, we basically varied it over a range from 0 to 1 and measured the deviation, the mean square deviation between the simulation results and the experimental data for each fret pair. And so delta is the mean square deviation between all the experimental measurements. And then we can calculate that delta as a function of the ratio of the coefficient of the charge interaction to the coefficient of the hydrophobicity interaction. And you see for most of the intrinsically disordered proteins, there's a minimum. So the smallest error between experiments and simulations, and it occurs around 1/2. So the hydrophobic scale is about 1/2 of the electrostatic energy scale. So another thing that we can do is measure a more global quantity, but also measure quantities that aren't explicitly fit for. So we basically, we have a fit or a best fit model for the for the models that I discussed before for each of the proteins. Then we can use that to calculate quantities that experiments didn't directly measure. So that would be the radius of gyration and the basically the radius of gyration as a function of amino acid separation. So I'm writing on my screen here, I have RG, the radius of gyration. So the radius of gyration is a quantity that determines the structure of not determines the structure, but it's a characteristic of the structure of a protein. So let's say I have some protein, and that's represented by that squiggle line in the lower right-hand corner. I measured the average amino acid position in the protein. So it's like, where's the center of mass of this protein? And what the radius of gyration does is it calculates the average radius of that protein. So you say that the protein is this glob, and there's a center of mass of that blob or glob, and then you ask, well, what's the average radius in all directions relative to that center of mass? So in mathematical notation, I have that formula that I just wrote for each C alpha atom, for example. You see how the position of that C alpha amino acid residue deviates from the average value, the center of mass. You square it, you sum it up over all amino acids, you divide by the number of amino acids, and then you take the square root. So it's a root mean square deviation of the structure from its center of mass. So overall, it gives you basically the size of a protein. So here I show you those values for different intrinsically disordered proteins that we simulated, both those that I talked about on the previous slide, but then some others that are in the literature. So HMG17, ATN, Pro-T alpha, the synuclein family, and then the tau protein. And you see that the model, which is in green here, agrees with the experimental values, which are in black. And then, as I've shown several times before, the electrostatic model, which is the diamonds with dashed lines, doesn't fit as well. And they're bigger, which makes more sense because if you just consider charge and no hydrophobicity, the structures will be extended. Now, the radius of gyration here is calculated over all amino acids in that structure. So it's like, that's why I label it RG sub NP, not sub NP, but RG of NP. That means I'm averaging over all of the amino acids in the structure. But you can also treat all the subchains of a particular protein. So I'll draw here. Here's an amino acid sequence going from amino acid number 1 all the way up to amino acid number NP. And so that's its full chain, but it has a lot of subchains. So for example, let's see, I have amino acid number 2 here. So there's a subchain on this protein, which is just amino acids 1 and 2. There's another subchain, which is amino acids 1, 2, and 3. And so you can talk about all the subchains and their length. So all of the subchains of length 1 are amino acids 1 and 2, amino acids 2 and 3, amino acids 3 and 4, so on and so forth. Subchains with length 2 are subchain 1, 2, and 3, subchain 2, 3, and 4, subchain 3, 4, and 5, so on and so forth. And then you could do subchains of length 3, subchains of length 4. And then there's only one subchain of length NP, which is the original chain. And so what you can do is ask, well, what are the radius of gyration or the separations for all the subchains? And that gives you a lot of data, which is really nice, and you can analyze that data. So it's the radius of gyration of all possible subchains given an original chain that goes from 1 to NP. And so that data is represented here. So instead of just talking about the radius of gyration of the full chain, I'm talking about which is the radius of gyration for something that has the full number of monomers. I'm talking about the radius of gyration for all the subchains that have length little n, which can go from 1 all the way up to the number of residues in each chain. And I plot that for a lot of different intrinsically disordered proteins on the left panel. So what you see is that the proteins have different what's called scaling. So I plotted this on a log-log plot. So if I have RG goes as n to the nu, then what you can do is take the log of both sides. So if the radius of gyration obeys the formula RG equals A times n to the nu, where nu is a a number. That's the new exponent, the new Greek letter is what's called a power law exponent. So if RG equals A times n to the new, then if I take the log of both sides, I get the log of the radius of gyration is equal to nu times the log of little n plus a constant, where the constant is the log of A, which is just that coefficient of that formula. And so if something obeys this equation, then it looks like a straight line on a log-log plot where the slope of the line is the value of nu. And so these lines roughly look like straight lines, and they have certain slopes. And those slopes are plotted on the right-hand plot as a function of distance from the, so I think on slide number 5, I had Q versus epsilon, and I had a line, sort of putative line that went from the lower left to the upper right. And so D is the distance from this line. So the idea is that it's the perpendicular distance. I haven't drawn it very well, but it's perpendicular. So the idea is that as you move to the left and up perpendicular to this line, you should be becoming more and more intrinsically disordered. And so the question is, does the exponent nu have any significant variation with the distance from this line? And so in general, according to polymer physics, the radius of gyration can go from basically the scaling exponent So the scaling exponent nu can have some typical values. So it can be 1 if it's basically fully extended. So if it's basically a straight line, the polymer is a straight line, then the exponent is 1. That's the limit of extremely charged extremely charged polymers. And then 0.6 is the limit of sort of uncharged polymers, but it's a excluded volume random walk. So the polymer's not collapsed in any extent, but it's not collapsing in any way. It's sort of fully extended, and it can't pass through each other. And then there's an ideal chain where it obeys Gaussian statistics. And so in that case, the exponent is 0.5. That's when you start to have some attractive interactions between the monomers in the polymer, and it starts collapsing a little bit. And then if it's fully collapsed, there's currently a debate in the community, which I'm a part of. It's how condensed can you get? If there's still some thermal fluctuations, people think that it's 1/3 the exponent, so it's really collapsed, but I think it can actually go lower, more like 0.25. And so those are the bounds of the vertical axis in the right panel. And so you see that if D is 0, so if you're very close to a folded protein, you're more on the collapse side. So maybe from 0.3 to 0.5, you're in the folded protein limit. But then as D gets bigger, as you move away from this separation between intrinsically disordered proteins and folded proteins, the exponent gets bigger, and that means that your charge is being is increasing. And so this may be a good idea, a good way to determine the likelihood that a given sequence or a given polymer will be a disordered protein and have a propensity to form aggregates. So with that, I'll just say that sort of the cutting edge now in terms of simulations is being able to put two, and two is just, the goal would be to put many intrinsically disordered proteins together to see if they aggregate, and to see when they aggregate, do they spontaneously form beta sheet structure, which is what people have been finding in the experiments. So currently, computationally, that's really difficult for models that are sort of atomistic or at the microscopic level. So I think cutting edge is thinking about how to speed up the simulations so that realistic length and timescales can be simulated and be used to predict aggregation. So some quick conclusions are that we built a coarse-grained C alpha model that seems to be able to capture the Fred efficiency, radius gyration of intrinsically disordered proteins. The agreement was sort of to within 10%, and the current bottleneck is being able to study multiple intrinsically disordered proteins to see what aggregates they form and whether there's a spontaneous formation of secondary structure. So with that, I'll conclude, and I'll stay around for a minute to see if there are any questions on these particular slides. Okay, see you on Wednesday.